Today we'll be studying about plant tissue culture, its principles and applications. So, hi everyone. My name is Joyson Paul, and let's dive into the area of plant tissue culture, how it is introduced, who are the scientists that are behind this plant tissue culture, and what are the principles they put forward, and what are the applications. So, plant tissue culture. So, we'll be studying the definition, history, techniques, regeneration, types of culture, nutritional needs, growth and maintenance, applications, pharmacognosy applications, edible vaccines, choice of explants. So we're gonna be deep dive into the all different things in plant tissue culture through the Mindmap. So let's look into the definition. So it kind of chunk it down into three parts. That is, plant tissue culture is to maintain and grow plant cells, its tissues and organs in sterile conditions and nutrient culture medium. So that is a definition of plant tissue culture, that it is used to maintain or grow plant cells, tissue organs, and maintain a sterile conditions. And also it provides nutrient culture medium. Next, let's dive into the history. So Gottlieb Haberlin's 1902 gives idea about the plant tissue culture, which is your totipotency proposal. Next, in 1930s, root tip culture, knocks in importance, has been introduced. 1960s, Morshige and Cook design medium has been introduced, specifically 1962. And standardize the nutrients, that what kind of nutrients can be introduced to grow plant tissue culture. 1970s, another idea came up with protoplast technology. And in 1980s, genetic modification or agrobacterium came up. In 1990s, expanded applications on cereals and legumes. So these are the history or background of plant tissue culture. Next, dive into the techniques. So there are 5 techniques. First is aseptic conditions should be required. It is use of laminar flow cabinet and surface sterilization by using or applying alcohol, hypochlorite. Next, your sterile containers, petri dishes and flask. Next is your temperature and light intensity. That is your control environment. Culture media, inorganic salts, organic nutrients, vitamins, plant hormones such as auxin and cytokinin, and gelling agents such as agar is introduced for the culture medium. And culturing techniques. These are all techniques per time. Next is your regeneration pathways. So regeneration pathways, we see pre-existing meristems, shoot nodal culture, organogenesis, non-zygotic embryogenesis. In types of cultures, how many types? So you could take seed culture, embryo culture, callus culture. Callus culture also, there are three types: undifferentiated cell mass, clonal propagation, and organogenesis. And organ culture, protoplast culture, cells without walls, genetic engineering, somatic hybridization. These are protoplast cultures. Next is your anther and pollen culture, that is your haploid plants, homozygous lines, mutation research. Next is meristem culture, virus-free plant, micropropagation. And the last one is cell suspension culture, which includes cells in liquid, agitated, secondary metabolite production, genetic transformation, etc. So these are the types of cultures that we use. Next is your nutritional needs. The nutritional needs include inorganic nutrients, organic nutrients, plant growth regulators, gelling, aging, optimal, optional components, natural extracts, MS medium. So inorganic nutrients includes macronutrients such as nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. And in micronutrients, it contains ferrous, manganese, zinc, copper, molybdenum, boron, and chloride. And organic nutrients such as carbon source, glucose, vitamins, amino acids. Next, growth and maintenance. So cell condition is required: control environment, regular subculturing, contamination, prevention, managing slow growth, managing necrosis. Applications: micropropagation, conservation, plant breeding, genetic transformation, disease elimination, production of sterile hybrids, artificial seeds, climate resilience, and induction of flowering. Let's look into micropropagation. It is exact copies of plants, rapid production, large number in reduced space, absence of seeds, regeneration of genetically modified plants, disease-free plants, germination of low germination seeds, reproduce recalcitrant plants. Conservation, rare endangered species, storage of genetic material. Plant breeding, screening cells for advantageous traits, protoplast fusion, embryo rescue, chromosome doubling, polyploidy, developing somaclonal variation. Secondary metabolites, pharmacognosy applications. Next, genetic transformation, testing genetic constructs, regeneration of transgenic plants, edible vaccines. Disease elimination, meristem tip culture, virus-free stock. That's all in applications. Let's dive into pharmacognostic applications. Secondary metabolite production, consistent quality, year-round production, enhanced yields, callus cell suspension, and examples includes Catharanthus roseus, vinblistine, taxis species, Orfolvia species, Coleus blumei. These are the example for application pharmacognosy. Next is edible vaccines. Genetically engineered plants, needle-free vaccination, transformation methods, examples and advantages. Let's look into transformation method, agrobacterium mediated, biolistic. Examples: hepatitis B antigen, cholera toxin B subunit, rabies glycoprotein. Advantages: ease of administration, reduced cost, and accessibility. Choice of eggplant, various plant parts, protopotential plant cells, meristemic ends, stem tip, axillary bud, root tip preferred, considerations, contamination management, and culture tissue challenges. What are the consideration? Growth and regeneration rates, haploid or diploid development, microbial contamination risk, semiclonal variation risk. Next is contamination management. It includes surface sterilization of explants, aseptically grown seedling as source, culture tissue challenges, slow growth, optimized medium responsive tissues, and necrosis responsibilities. So that's all in plant tissue culture principles and applications. This is basically a mind map to learn all those things easily. Thank you.